Hi, my name is Mark Kolbeck, and I'm going to be presenting to you a series on cardiac electrophysiology. There's a fair bit to this series of lectures, and I'm going to try and keep them short, about 10 to 20 minutes each, so that you can take them in small bite-sized chunks. And I've learned over the years, I've been teaching electrophysiology now for over 10 years. I've learned that sometimes people don't always have the fundamentals I assume that they have when I do this series of lectures. So I'm calling this one Part 0, Fundamentals. I also want to say at the beginning of this series, though I won't say it through the entire series, that all the content in this series is available under Creative Commons Share and Share Alike license. I'll put the lectures up on YouTube, link the lectures to my paramedicine.com website, and I'll put the slides up on the paramedicine.com website. So if people want to download the slides and use them for your teaching, as long as you are not using them for a for-profit purpose, then I'm happy to share these with the world. My philosophy is that I was very, very fortunate that I've been taught by some really fantastic people. And I know this information, without overstating it, reduces suffering and saves lives. So I feel like it's part of my moral duty to be able to pass this on to the next generation so that you guys have this too. As long as you don't use this for profit, it's yours. It's yours to use. So let's talk a little bit about some of the basic fundamentals before we start really getting into cardiac physiology. This is what we're going to talk about in this first lecture, Lecture 0. We're going to talk about basic building blocks of life, of humans, of cells. We're going to talk about cells and cell membranes. We're going to talk about what's inside cells, and we're going to talk about important ions. Because here's the thing. If you really want to understand cardiology, if you really want to understand what's going on in the heart, if you want to understand dysrhythmias, if you want to understand ST elevation and all this stuff that's going to come later, then you have to understand ion. When I mark a student's paper and they say something like, oh, the heart beats because the electricity goes back and forth, they're getting down to the molecular level, but not quite. I want a clear understanding of what is going on at the molecular level. And if you understand that, the rest is easy. If you really take the time to build this foundational building block of knowledge, that cardiology is all about, electrophysiology of cardiology is all about the movement of ions, then you can build everything on top of that solid structure. So that's what we're starting with. And if this is really simple to you, as it probably would be if you're a university student, that's great. It's material you've already covered. But if you're coming into this and going basically from 0 to hero, and you want to start at nothing and learn to interpret 12 leads for STIACs, then this is important foundational information. So basically what I'm saying is, forgive me if you already know this. And if you don't already know this, then you're welcome, because it's really useful stuff to know. Another thing I'm going to do through the series of this lectures is start with priming questions. I'm a scientist, so I try to do everything according to the evidence base. And the evidence base shows that when you're giving a lecture, when you're presenting information, first of all, you should never go for more than about 20 minutes, because then people start to go and their brain melts. And second of all, if you introduce a bunch of questions at the beginning without giving answers, then that primes your brain to start thinking about, oh, wait, that was one of the questions at the beginning, and you're more likely to remember it. Also, after this lecture is over, you can go back and look at the priming questions, write them down, and write down the answers to them right after the lecture, which I would strongly encourage you to do if you're really trying to learn and retain this stuff. And that will help reinforce the information. So here are the priming questions. First one first. What's an element? What is an ion? It's a bit different than an element. When we're talking about ions, what about an anion and a cation? What are those? They're subdivisions of ions. What is a cell, and where did they come from? And what are cells made out of? And what are the major elements, the major building blocks that human beings, that you and I and dogs and all other life are made out of? And when we're talking about those elements, why is phosphorus in particular so important? And then finally, I think on this slide, what are the 3 specific ions that we're going to be talking about through all these lectures as we talk about the electrophysiology of the myocardium, of the heart. So, elements. Elements are basic building blocks of the world. When the Greeks tried to figure out what the world was made out of, they could break it down into, large things like fire, air, water, earth, that sort of stuff. But they wanted to break it down to the most indivisible parts, what are the smallest building blocks that we know of in life. And they called those things atoms. And atoms in the Greek means basically indivisible. So the atoms are synonymous with elements. The elements of the atoms. Elements are the indivisible things that make up the world. Or at least that's what the Greeks thought. We've since found out that there's stuff that makes up the elements, but we're not going to get into that. We're going to talk on a relatively large scale in terms of molecular chemistry and just say that the world is made up of elements. And it doesn't look like that. If you were, for example, to look at a huge tower like this, you'd think that is one big monolithic thing. If you look at a human, if you look at an elephant, you think that's a thing. But we know that those big things are made up of smaller building blocks. And a big tower like that is actually made up of little tiny Lego blocks. So if you think about elements as the Lego blocks of the world, these tiny, submicroscopic, teeny things that make up the world, then that's a really good understanding of what elements are. And most of you are familiar with the periodic table of the elements that lists all of the elements that we know. And I'll show you the periodic table as we go through this. If we keep using this Lego metaphor, then imagine that you can take some of those Lego blocks and put an electrical charge on them. So you get like a static shock when you touch them. And that makes some of them stick together and some of them repel. So you can put in electrical charges into these elements. The way we do that is by taking a part of the element called the electron. So elements are made up of electrons, protons, and neutrons. We take some of the electrons away. And if you take away an electron, which has a positive charge, then the Lego block that's left over has a slightly positive charge because you've taken some negative away. If you stick some extra electrons onto that Lego block, onto the element, then it gets a negative charge because electrons are negative. So you've added more negative, made it more negative. Well, when we talk about an element that has a charge, an element that has a charge is an ion. So elements and ions are basically the same thing. It's just that an ion is an element with a charge because the electron's been taken away or some have been added. If you have a positively charged ion, that is a cation. If you have a negatively charged ion, that's an anion. So those are some basic terms that I hope you're already familiar with. If you're not, sit down for a second, write them out on a piece of paper and make sure you're comfortable with those and you've got the definitions with you while we go through the rest of this lecture. So, what are the Lego blocks of life? If we take a look at the Lego blocks of dogs and cats and humans and everybody else, there's basically 4 major elements that make us up: carbon, hydrogen, oxygen, and nitrogen. And if you took all of the elements of a human being and broke them into individual piles, over 99% of our body by weight is carbon, hydrogen, oxygen, or nitrogen, which we sometimes call CHON. So we're basically CHON. We're made out of CHON. If we were Lego structures, there'd be 4 bricks that would make us up, or make up 99% of us. That would be the carbon brick, the hydrogen brick, the oxygen brick, and the nitrogen brick. That's what I am. That's what you are. Less than 1% of that is some phosphorus, sulfur, sodium, magnesium, chloride, potassium, calcium, and iron. So there's other trace elements. There's a little bit of spices in us. But mostly, you are CHON. That's what you're made out of. So if you take a look at the periodic table of the elements, this is a listing, mostly, of the elements that we know. And so you can see some of the major ones if you take a look up here: hydrogen, sodium, magnesium, potassium, calcium, a little bit of iron, carbon, nitrogen, oxygen, phosphorus, sulfur, and chloride. So chlorine. This is what makes us up. This is the stuff that you are. So in blue there is the CHON, and in yellow are the other trace elements, less than 1%, that actually make us up. And they're all stuck together somehow to make humans and dogs and cats and snakes and everything else. Somehow we take these elements and build them into living things, which is amazing. So phosphorus is a particularly interesting one that we'll talk to because the way we build our body up is to take these elements and to build them into cells, and then those cells make up the building blocks of our body. So elements make cells, and cells make bodies. And phosphorus, even though it's not one of the big four, the cation, is really important in the construction of cells. Without phosphorus, we don't get cells, and without cells, we don't get life. So phosphorus becomes really important. What is this stuff? Well, surprisingly, when we find it in nature, it's really reactive. It reacts with other stuff. In fact, it's so reactive that we use it in explosives. Napalm is mostly phosphorus. It can go into poisons. It can go into nerve agents. There's a type of fertilizer that we use called organophosphates. But they can also be used as neurotoxins. If humans get poisoned with organophosphates, we run into real trouble. It was the 13th element. So there's hundreds of elements that we've discovered. This was the 13th that was discovered, and it was discovered by distilling urine. They used to take like liters and liters and liters of urine. It takes 1,000 liters to make 50 milligrams of phosphorus. So you can imagine what those old production facilities were like, pretty gross. Later on, we figured out a way to get it out of bones, and now we mostly get it out of rocks. So when we use phosphorus industrially, that's how we get it from. That's how we get it. But in our bodies, we don't go, eating rocks or eating other people's bones. Well, sometimes we do, bone broth. But we need to have phosphorus inside of us because it's an important part of our DNA, which is the protein structure that makes up our life, and RNA, which is another protein that helps create DNA, and adenosine triphosphate, which is the basic battery that we operate our body on. And it's also really important, and this is where it comes into this lecture, it's really important in something called phospholipids. So phospholipids mean we take this element, phosphate, and we attach a lipid or a fat to it. So imagine that you've got like a little hand floating around and you're just glomping some fat onto it. That is a phospholipid. Why are phospholipids so important? Here's why. If you take phosphorus and you bind it to 4 oxygen elements, so 1 phosphorus and 4 oxygen elements, we get something that is hydrophilic. And philia means to love. So when you take a phosphorus Lego block and 4 oxygen blocks, you get a little thing that's charged and it really likes water. It absorbs well into water. It doesn't repel water. But then when you take the fat and you attach it to the phosphorus, and fat is a whole bunch of blocks of hydrogen and carbon Lego blocks. So you take phosphorus and 4 oxygens, and you take carbon and a bunch of hydrogens, and you smush those 2 things together. And you get something that kind of looks like this, which is called a phospholipid. And the phospholipid has one region that likes water and another region that repels water, because it's mostly fat. If you imagine pouring like hot bacon fat into a sink full of cold water, it doesn't mix in nicely the way sugar or coffee or something like that would. It forms into little globs and it repels the water. They don't mix. So these phospholipids, phosphorus, oxygen, carbon, hydrogen, make up these little phospholipid things, and they're really, really important. And here's why. Because when you take a bunch of phospholipids and just throw them into a sink full of water, what happens is the fat, which doesn't like the water, goes into the middle here, and the heads, which like the water, form on the outside, and you get phospholipids forming 2 layers of phospholipids coming together. If this is one phospholipid layer and another phospholipid layer, this is the part that loves water. The back of my hand loves water. The inside of my hand hates water. So throw them into a big vat of water and they go wonk, like that. And the part that hates water is hidden in the middle, and the part that loves water is exposed to the water on the outside. That's called a phospholipid bilayer, two layers going together. And it doesn't take any magic to do this. Throw a bunch of phosphorus and a bunch of hydrogen and carbon. Just throw those Lego building blocks into a sink full of water, and they will start forming phospholipid bilayers all on their own, just because of the attraction and the repulsion to water. Now, the problem with this, obviously, is that if you take a look here, this stuff is still all exposed to the water, and this is hydrophobic. This is water hating. So how can you make this middle section here not exposed to water? Well, the trick is you turn it into a three-dimensional sphere. And if you throw in phosphorus, oxygen, carbon, and hydrogen into a big vat of water, they automatically form into spherical phospholipid bilayers. And that is called a liposome. Lipo means fat. So a fat body, a fat thing. And this, for the first time in sort of spontaneous creation of stuff, is in the history of the Earth when we had things that had an inside and an outside and a layer separating the inside and the outside. So liposomes are really important because evolutionarily, they were the beginning of cells. And cells are just fancy liposomes. That's all they are. So get a whole bunch of fat and some phosphate and all those ingredients, throw them into the ocean, and they will start to form little spherical fat blobs with the ocean on the outside and the ocean on the inside, but the two are separated. Very important evolutionary step. So what's inside of liposomes? Well, if you take a look at the constituency of seawater, you've got a bunch of water and you've got some other important components in there, most of which are ions. And there's a breakdown of the ions that are inside seawater. And not surprisingly, that's the same thing that's inside most liposomes. If you ever hear biologists saying we carry the ocean inside us, that's what they're talking about. These liposomes formed inside the oceans. They formed into cells in the oceans. Those cells gradually evolved into life in the oceans, and the insides of those cells are a lot like the outsides were, the ocean. So what's inside those seawater? Chon and some trace elements. What's inside of us? Basically the same sort of stuff. So seawater doesn't have as much nitrogen, phosphorus, and iron, but what our liposomes eventually learned to do was they learned to take in some things that they wanted and to expel things that they didn't want. So the liposomes started to become active as they turned into cells and started to be able to select the environment that they had on the inside and make that environment slightly different than the stuff on the outside. So if we take a look at single-celled organisms, living beings that are essentially just one liposome, we can ask, well, what's inside those single-celled organisms? And the answer is basically the same stuff as seawater. So some sodium, potassium, magnesium, calcium, chloride, bicarbonate, sulfate. So that is very similar to the ocean, which is why biologists talk about we carry the ocean around inside of us. And it's why when you cry salt water, because we're carrying the ocean around inside of us. So as single-celled life started to become multicellular life, they started to create bodies made up of cells. And in between the cells, the interstitial fluid, that fluid between the cells was again very similar to the fluid that's in the ocean. So the insides of the liposomes that have become cells is ocean-like, and the space between the cells in multicellular organisms is very ocean-like as well. So as this evolution happened, as life from the ocean started to move into life on land, it retained its internal environment. It retained the same sort of concentration of ions and anions inside the liposomes, inside the cells, and surrounding the cells. So we took the ocean with us. When we left the ocean, or say life, I didn't, but you didn't, but our genetic predecessors, the organisms that left the ocean and came onto land, they took the ocean with them onto the land. So when we look at cells now, we say that there's extracellular fluid, which is also sometimes called interstitial fluid if it's inside a body. So if you've got a bunch of cells inside a body, the extracellular fluid is sometimes called interstitial. And you've got inside the cell a bunch of fluid. Cytosol is what we call the stuff inside the cell. And again, very, very similar. Not entirely same concentrations, but the same basic elements, the same basic ions and anions. But here's the problem. Once you create a liposome and once it turns into a cell and you've got this phospholipid bilayer, it's difficult for some things to get through there. Very small elements, especially non-charged elements, elements that aren't anions or cations, they can get through fairly easily. So oxygen goes right through the phospholipid bilayer, no problem. Carbon dioxide goes through it very easily. Small fats and hormones, some anesthetic agents, water can move through very easily. There's no special mechanisms required. But once an element becomes an ion, once it gets a positive or a negative charge, or if the element turns into a bunch of elements clumped together, which we call a molecule, like proteins or glucose, then those things can't get through the phospholipid bilayer unless they get some help. And we'll talk about the help. We'll talk about the different channels and pathways through the phospholipid bilayer in another lecture. But right now, the basic thing that I want you to understand is that if you throw all these elements into a sink, they form together into a phospholipid bilayer. That's just automatically what happens, which we call a liposome. And then as that liposome starts to get energy, gets some mitochondria inside, and starts to get some other structures moving inside it, and starts to become a little bit more sophisticated, then it can start to control its own internal environment. Think about it this way. You throw a bunch of tires and metal and some glass into a great big industrial vat, and a car just automatically forms. That's like our liposome. But in order for that car to actually get out and start moving, you got to get an engine, you got to get an exhaust system, you got to get some fuel, you got to have a way to get the fuel in, you got to get a way to push the exhaust out, you got to have a mechanism for that engine to be working. That's when a liposome turns into a cell. So a liposome is basically just the shell, just the structure. A cell is a liposome that has a whole bunch of internal mechanisms inside of it so that it can actually move around, so it can control its internal environment, so it can create new cells. And that's when the shell of a liposome turns into a live organism, like a single cellular organism. So as we talk about the ingredients inside the cell, there's a bunch. There's all sorts of different things, but there's three particular ones that I want you to know about, and those are sodium, calcium, and potassium. And all of these three have a positive charge, so they're cations. So sodium has one positive charge, calcium has two, potassium has just one. And you'll notice that calcium is really big, potassium is really small. So this is the sodium, calcium, potassium. Potassium is really small, calcium is really big, and sodium is the Goldilocks right in the middle. And when we're talking about for example, nervous tissue, then we don't worry about calcium. Nervous tissue is just sodium and potassium moving back and forth. But when we're talking about muscle, calcium becomes really important as the driver of contraction in muscle. So as we talk about all of the things that we're doing in cardiology, if you want to know if you really understand it, the trick is to go back and say, how is the movement of sodium, calcium, and potassium causing this thing that I'm thinking about? And if you can do that, if you can always bring down whatever topic you're talking about all the way back to sodium, calcium, and potassium, when we're talking about the heart and contraction and electrophysiology, then you really understand it. You really understand what's going on. So make sure you understand these cations, you know what they are, because we're going to be talking about them a lot. The reason we've got this color, by the way, is just when you react the different elements with a flame, that's what they glow with. So I'm adopting that sort of coloring as I go through these lectures. So that, I had to color them something to make them distinctive. So I thought I'd just go with what happens when you actually burn them in a light. This also explains why different streetlights are different colors, because they're actually burning different cations inside there. Okay, that's the first lecture. If you have any questions, then feel free to list them underneath. Take a few minutes to reflect on this, make sure you understand all the material, and then we'll move on into actually lecture 1, which is moving beyond here and just establishing the fundamentals.